Hi everyone, I'm Hannah and we are the Polypeptide, Helicity, and Ensemble prediction tool team. And now I'll introduce the rest of the team, or as we like to call ourselves, the PEP squad. So going from left to right, we have Marco, Eric, Amy, Samia, Jonathan, and myself. And PEP squad is short for peptide squad because, to put it simply, this tool predicts peptide structures. And over the past several weeks, we have immersed ourselves in the biochemistry of this project. And now I'd like to give you all a quick rundown of the scientific background. So you might be wondering, what is a polypeptide helicity and ensemble prediction tool? Well, Dr. Terry Oss and Dr. Scott Schmidler, collaborators between the Duke Biochemistry and Statistics departments, have developed a tool using Bayesian statistics that can predict the helicity of any peptide sequence. And helicity refers to the alpha-helix structure on the bottom left, and a more helical structure is more rigid compared to the coil structure on the right. So researchers can use this helicity coil information to design better antibody linkers that have the adequate flexibility to target proteins responsible for diseases such as HIV, COVID, and cancer. And now I'll let Amy talk more about the capabilities of this polypeptide helicity tool.
Thank you, Hannah. So the first function of this tool is the ability to calculate the average helicity value of the entire polypeptide sequence. The input values for this function and the two other functions of this tool are sequence, temperature, pH, N block, C block, urea, and TMAO. N block and C block refer to the charges on the polypeptide endings, and urea and TMAO refer to the concentrations of those two respective compounds. And the data frame on this slide is a screenshot of what the tool outputs. As you can see, the average helicity of this sequence was roughly 14%. The second function of this tool is the ability to calculate residue helicity. That means that instead of calculating the helicity of the entire polypeptide sequence, this function goes through every single residue, every single amino acid of the sequence, and calculates the helicity at every single point. The diagram on the right is a zoomed in version of the data frame on the left. The residue column refers to the indices of the residues, and the helicity column contains the helicity percentages of the corresponding amino acids. The most advanced feature of this tool is the ability to output a list of possible configurations of the polypeptide ranked in order of descending probability of occurrence. And there are 2 to the n configurations for a sequence of n length. This means that if someone is submitting a sequence of 20 amino acids, that's around 1 million different configurations that this model outputs. And instead of forcing the model to return the maximum possible number of configurations every single time, which would take up a lot of memory and runtime on the back end, the user is allowed to specify the number of configurations they want to see. Along with each configuration, the engine also outputs information such as average helicity, statistical weight, and free energy. 9 weeks ago, our team was given a GitLab repository with research-grade C++ code and instructions on how to create a file in a terminal and run the model. But that was far from ideal for the researchers who want to use this model. So our team developed a web application that allows researchers and our stakeholders, collaborators to run the model through a website. And next, I'll pass it along to Eric to speak more about the tech stack of our deliverable.
During this project, we were able to get familiarized with many software development tools, with our main front-end tool being Jupyter, and we used Fast API to dock to the backend engine, as well as PostgreSQL for our database. Next, I'll talk about our system architecture design. This is a diagram of the system architecture we decided to implement. How it works is that the user inputs information about the polypeptide to the public interface, which is then transferred to the backend Helicity Ensemble prediction engine by making a Fast API endpoint call. The output of the backend is then sent back to the user interface as well as to an SQL database, which the admin page directly pulls from. Furthermore, the admin interface also works concurrently with an AWS S3 offsite storage. Now I'll let Samia talk about the components of the public interface.
Thank you, Eric. So currently you guys are looking at our start page. We have 3 functions that the user can choose between based on the information that they want to know about their polypeptide. Above these buttons, we have descriptions of the functions as well as icons to represent each one of the functions. Additionally, we have a navigation bar, so the user can navigate between the home, about, and FAQ page. So once the user presses a button based on the information that they want to know about their polypeptide, it will then bring them to this page where they have optional inputs such as name, email, and institution, the option to upload multiple polypeptides as a file, or the option to just upload one polypeptide. So we have our required input, sequence, temperature, and pH for the polypeptide to be accurately predicted, but we also have optional inputs such as N block, C block, urea, and TMAO concentration. So we have many ways that we handle errors on the front end. So an example of an invalid sequence will be any sequence that has the letters B, J, O, U, X, and Z in them. If we take a look at the next slide, if we look at the file section that says N block and C block, we see that the user has entered 0, which is not a valid input for N block and C block. Below that, you see the red error message the user will get, letting them know what directly is wrong with their inputs. Now I'll let Jonathan discuss the components of the admin interface.
This is the admin page, and here we can query through the entries. And for the input and output, we are using Amazon S3 to be able to create URLs, which we can open and install. And we also can see the frequent users as well as the memory use. And now I'll pass it on to Marco to speak on authentication.
Thank you, Jonathan. So one of the features of the admin page is, as we mentioned, authentication and authorization. This way, only admin users such as us, the project team, and our stakeholders can access the page. And here we can see a very simple diagram of how authentication authorization is implemented. And as with most Duke-affiliated sites, admin users are prompted to log in with their Duke credentials, and if authorized, they'll be redirected to the admin page. With this said, I'll go ahead and pass it off to Eric to talk a little bit more about how the backend works, and specifically how it handles errors.
So to ensure proper communication between the various components, the backend also needs to check for errors. Other than the standard connection errors, we also made our own internal service errors to check JSON format and runtime errors to make sure there's no input that causes an unpredictable massive runtime and memory usage. For example, as shown in the top right, if we run into a runtime error, we send the current information we have to the database so admins can see what error occurred, as well as information about the user who ran into that error in case the admins would like to contact them. We have also implemented CI/CD, or continuous integration and continuous deployment. And we have two branches, main and development, hosted on mine and Hannah's virtual machines respectively. And we've recently added a third branch, production, where it will be deployed 24/7 and available for users to access. These three branches are completely separate and allows us to make edits to specific branches without messing up other branches in the process. Now I'll pass it on to Marco to talk about some testing we did with runtime and memory.
Thank you, Eric. So the public interface team ran performance tests of peptide lengths against runtime and memory, and this was done in order to determine the hardware resources needed to run the polypeptide Helicity tool. And to be more specific about how we tested the tool, the team tested around 50 different polypeptides, about 3 times each, for a total of 150 tests. And just for some technical context, the tool was tested on a virtual machine that has 4 core processors and a base memory of 16 gigabytes. And from the graphs above, we can see that both runtime and memory start to increase exponentially at around 50 amino acids with some variability between the runs. And from these tests, we can conclude that the virtual machine can in fact handle running the tool, and running the tool on a production server or virtual machine for that matter is sufficient for the needs of our web application. I will now pass it off to Hannah to go over the demo of our web application.
All right, so here is our homepage where the user can choose one of the 3 functions that our engine provides. I will first show you the calculate average Helicity function, where the user is brought to this input page and they can fill out some optional contact information. While we encourage them to fill this out, if they would like to remain anonymous for research purposes, they can opt out of this. And once they fill out that contact information, they can then submit one or multiple peptides. And in this case, the user is going to submit multiple peptides. They do this by uploading a CSV file. And once they upload that CSV file, they can hit that submit button. And the output is the average Helicity calculation for each of the sequences that they inputted in that file. Now I will show you the calculate residue Helicity function, where again, the user is brought to this input page and they can fill out their contact information if they choose to. And once they fill that out, they can then either submit one or multiple peptides. In this case, they're submitting one. So they hit that submit button and they get a graph showing the helicity calculated at each residue, as well as a downloadable table with those helicity calculations. And a new feature that we are still testing is the ability to visualize the sequences that a user inputs. So this is a 3D model of that sequence, and we're able to drag it, we can zoom, and it is still a work in progress, but we wanted to show that to you today. And finally, we have our enumerate ensemble function, where the user is again brought to this input page, and they can fill out their sequence and other inputs, and they can set the max probability to limit the data that is returned. So hitting that submit button, the user gets helicity and probability graphs, as well as a list of ranked conformations for the sequence that they inputted. And the C's represent coils, while the H's represent helices. And you can click through that if you choose to, and also download it if you would like. And finally, I'll show you our static pages. So here we have our about page, which goes a little bit more into the science behind this project, as well as how the model came to be, and also recognizing our developers for this project. And you can also see our FAQ page. So if a user is unsure about what exactly is considered a valid input for one of the experimental conditions, they can check out our FAQ page and click through any of the frequently asked questions to get some more information. And they can also download our template if they want to upload a file with multiple peptides. Now, if we hop over to our admin page, here the user can query through our database with these search boxes and apply those filters, which pulls log entries from our SQL database and displays it to the admin page. And we can also click through that, and we can also open input and content files. So if we open that input file, we can see what a user submitted to our engine. And we can also open a content file of that exact submission and see what a user's output was for that particular submission. And another feature we've added is the ability to download a full CSV of the displayed log entries. And here it will show all of the log entries from our database. We also can track user traffic. So we have our top 5 users to date, and we also have our memory usage over time graph. And now I'll let Marco talk about our future steps.
As you can see from the demo, we've come quite a long way since the first week of the program. With that said, however, here we have some of the future steps that we envision the project taking. So for example, one of the steps we have in mind is enhancing the 3D visualization of the peptide structures and upgrading the security measures on the website itself. Finally, to wrap up this presentation, I'd like to end off with some acknowledgements. So first and foremost, I'd like to thank our team leads, Mark McCahill and Jeremy Bandini. I'd also like to thank our stakeholders, Terry Oos, Scott Schmidtler, and Roy Hughes, as well as to our corporate sponsor, Mark Laverne. Last but not least, I'd like to thank the CodeLab, as well as to our program facilitator, Jalen Cuellar, and the rest of the Code Plus team.